right. Okay, oh, I forgot I gotta start my own time. Okay. Before I start my timer, I will probably go a little long because I still need to practice a lot, but okay. All right, hello everybody. My name is Nathan Williams. I'm part of the PelWAD laboratory, and today I wanted to discuss the enhancement of adeno-associated virus gene delivery in cells by cationic amphiphilic drugs. Okay, so I first wanted to introduce adeno-associated virus as a gene therapy vector. So that means AAV is used to basically deliver beneficial genes to target cells and tissues for a therapeutic purpose. So one of the great strengths of AAV for this gene therapy is its versatility. So basically, the AAV gene therapy vector has a few different serotypes that have different affinities for different tissues. So you can see here, AAV has been adapted to different types of disorders and different types of tissues. And these are all clinical trials of AAV gene therapy. So on the left, you can see a very simplified structure of the AAV capsid. And so the reason this is called recombinant AAV is that it's been inserted with a custom gene. So usually this will be the therapeutic gene that you're trying to deliver to some tissue. And you usually put some kind of promoter that will express that in any cell or in a specific cell. You can also see this PLA2 domain, this phospholipase A2. So that just means, the name just means it cleaves lipids and it helps the AAV capsid to reach the nucleus in the cell. Okay, so the main problem I want to solve with the AAV gene therapy is its cost. So you can see here I have an example of one of the approved AAV gene therapies that was actually withdrawn after 2 years in the European market. It was over $1 million per treatment. So you're trying to treat one person with AAV, it'd be over $1 million. One of the 2 FDA approved AAV gene therapies in the US is over $2 million. So it's pretty expensive. And the reason for that is AAV's transduction inefficiency. So you can see in red, I've highlighted the degradation points. So there might be other pathways that they haven't identified that result in inefficient transduction, but essentially AAV capsids are taken up by the cell through endocytosis, and there's a chance they get degraded in lysosomes or degraded by proteasomes. But any of the infectious AAV, they escape from late endosomes and lysosomes with their PLA2 domain. So the way they escape is by the increased acidity of these late endosomes and lysosomes, which activates the PLA2 domain. So any of those capsids, there's a chance they reach the nucleus and deliver their gene to the nucleus. So they disassemble in the nucleus and deliver that way. But there's also a chance they degrade. So my goal is to enhance the transduction efficiency of AAV capsids by improving this escape step. So we're hoping to bypass both of these degradation steps by overwhelming how much capsid reaches the cytoplasm. And then we can enhance gene delivery. So overall, I want to enhance AAV efficiency, reduce the required dosage for treatments, and reduce the cost of AAV treatments. So how will I do this? I aim to use cationic amphiphilic drugs. Okay, so before I talk about the definition of CADs, I want to discuss how they move through the cell. So essentially, what I want to exploit about CADs is that they destabilize late endosomes and lysosomes. So the idea is to assist that PLA2 and hopefully get some kind of synergistic destabilization of these compartments. So you can see that CADs will diffuse through the membranes of the cell and they end up accumulating in more acidic compartments. So you can see as the pH drops, you will see an increase in CAD concentration. Okay, so now I can explain why that happens in the cell. So here you can see CADs share certain structural properties. So they're small, amphiphilic or lipophilic partially. They have basic functional groups and they're a pretty diverse group overall. So you'll have antipsychotics, anti-allergens, antidepressants that are all, they all happen to be CADs. So this is one example of a CAD being protonated, and that's an antidepressant. Okay, so because of the CAD's small size and their lipophilicity, or their hydrophobicity, so that's what a log P will mean. It means it's able to mix with fats or hydrophobic mixtures. They are able to pass membranes, and when they reach a more acidic compartment, they become protonated, right? So once they reach like a pH of 4.5 or maybe 6, they will become mostly protonated and charged, and they can no longer cross the membrane of acidic compartments. So that is how they accumulate, like I showed on the left. And I wanted to note what also assists this accumulation is the binding of negatively charged lipids within these compartments. Okay, so overall, I hypothesized that CADs will enhance AAV transduction by synergistically disrupting late endosome and lysosome membranes and enhancing escape into the cytoplasm of these capsids. So then we hope to see enhanced gene delivery from this synergy. Okay, so in my first aim, I'm hoping to establish structure-activity relationships between CAD structure and these enhancement activities. In aim 2, I want to take a closer look at the membranes and see how CADs and AAV capsids work together to, or don't work together, to destabilize these membranes. Okay, and then in aim 3, I want to see how does the location change when we add CADs of AAV endocytosis. So normally they'd escape in certain locations. We want to see if the CADs support this or if they cause escape at a different location in the cell. Okay, so for my first aim, so I'm looking to establish structure-activity relationships for the CADs, and that way we can predict better which CADs have activity with AAV. So here I used a recombinant AAV with an eGFP gene. So that means if the cells get transduced by AAV, they start to synthesize GFP, right? So this is a control. You can see there's not too much GFP synthesis, and this is after 24 hours of co-incubation. And carvedilol, this CAD here, also doesn't show a huge change. But you can see with some of these CADs, we see a fairly dramatic change of 2 to 3-fold. So the mystery here is what's causing these differences. Why is it that carvedilol is not great for enhancing AAV transduction, while these others seem to show some kind of enhancement? Okay, so I want to start by selecting a diverse library of CADs. You can see all these different drug classes that they're part of, antiestrogens, antimalarials, and they all have these different pKa values and log Ps. So they all have the properties of CADs, but they have slightly different variations of pKa and log P. And they're all pretty small as well. So overall, I'm trying to see how the structural properties of these CADs informs AAV transduction enhancement. Okay, so here we can see the system I'm kind of working with for aim 1. So I'll be mixing recombinant AAV and cationic amphiphilic drugs. And once the AAV are in the cell, we should see some gene products synthesized. And overall, I want to define correlations between properties and enhancement. So I'll define or quantify activity in 2 ways: how much gene product is expressed in the cell from infection, and how quickly does AAV reach the nucleus, and how quickly does it reach the cytoplasm. Okay, so to see how much gene product is expressed, I'll use a recombinant AAV that, again, if there's transduction, we should see synthesis of GFP in our cells, right? So then we should have fluorescent green cells, or some population of them, and this will represent our transduced cells. So then I can use flow cytometry to identify the transduced population. So I can see which cells have a higher amount of EGFP, and then I should see this fluctuate with and without CADs, right? And that way I can quantify how much CADs affect transduction, and I can also image for qualitative images like before. So that's the system I used on the previous slide. Okay, so I also want to look at how quickly does AAV reach the nucleus and the cytoplasm. So I will label capsid with Alexafluor 488 and make the capsid itself fluorescent. So when I do this, I can incubate with the fluorescent capsid and CADs, and I can observe the differences in the capsid localization. So I can use a membrane stain to stain the membrane and define the boundaries of the cell, and I can use a nuclear stain to see where AAV co-localizes, right? So that will tell me where AAV has entered the nucleus, and I can compare these localizations across each CAD. So I would theorize that the CAD enhancing, or the AAV enhancing CADs should show a more intense signal throughout the cell at a faster rate. Okay, so moving on to the second aim, I want to take a closer look at the membranes that are being disrupted by AAV and capsid. So at the level of the late endosome, we expect, or we know that AAV capsids escape by disrupting the membrane, but we don't know exactly how, hence the fairly vague blip that I have on the membrane here. But we do know it escapes somehow with the PLA2 domain, and we also know that CADs should be binding with late endosome and lysosome bilayers, and that's because of negatively charged lipids there. Okay, so I have two questions. How do CADs and AAV together cause this disruption? Do they enhance each other to give us a synergy? And then how does CAD binding correlate with this disruption? Okay, so I'm not going to look at this in the cell. So I'm going to take a step out to in vitro and use large unilamellar vesicles, a type of liposome, and I can change the composition of these liposomes to match either the early endosome or the late endosome. Okay. So to measure the amount of disruption by CADs and AAV, I will load the LUVs with calcine, which is a fluorescent molecule that's quenched when it's in a very tight space. So we shouldn't see any fluorescence when the calcine is in the LUV, but upon any disruption from CADs and AAV, we'll see leakage and fluorescence. Right, so then we can quantify this by size exclusion HPLC and compare that among different CADs. And we can also change pH and, of course, the membrane composition. And I also want to see how CAD binds to lipid bilayer by using fluorescence anisotropy. So on their own, CADs will rotate and tumble very fast. And as they fluoresce, they'll disperse the fluorescence signal all around. So we input a polarized fluorescence signal, and we should see that polarization get lost because the molecule is moving so fast. So if we add an LUV and we see any binding, we should see a slower rotation, slower tumbling. So that means we're retaining the polarization more from our fluorescence input. So we put fluorescence in here, we should see we don't lose as much of the polarization. So that's how we can measure binding using anisotropy over a change in concentration of CAD. Okay, so then that would let us determine how well do CADs bind to membranes or lipid bilayers, and how much do CADs destabilize these bilayers with AAV. Okay, so now moving back to the cell. So I want to determine how CADs modulate when and where capsids enter the cells. So basically, for this first part of this aim, I want to use inhibitors of endocytosis and see where the CADs can overcome this inhibition and see where the CADs impact endocytosis. So again, I'll use a fluorescently tagged capsid, And these, I've just marked these with numbers, these different steps where our inhibitors might be able to inform us about escape. So one example is if we inhibit Rab7 here, we'll be looking at the early endosome step. So if we inhibit that and we add AAV and CAD in image, and we compare this to just AAV alone, if we see a significant change in the amount of AAV in the cytoplasm and the nucleus, we might say that CAD impacts early endosome escape, right? So if we saw that CADs can change this even when this is inhibited, that means the CADs are impacting the step. Okay, and that's just one example. Okay, so another assay here to look at endocytosis. I want to see how CAD-induced damage to these membranes informs transduction. So essentially, I want to use a damage response protein that's been tagged with a fluorescent protein, or a fluorophore, and see how this responds to CAD disruptions. And I can use other constitutive endocytosis proteins to see where this protein's moving by co-localization. Okay, so here's a little example. So I don't think you can see it very well. I might need to boost the contrast. But there's a vague or a dispersed red signal from our Gal9, and over 24 hours it shows no puncta at all. But if we add chloroquine and we wait 8 hours, we'll see puncta start to form as this Gal9 localizes to different membranes. And the nucleus is stained with blue there. Okay, so overall, I wanted to summarize the expected outcomes. We will gain knowledge of how CAD properties predict changes in AAV transduction, and we'll learn which CADs are most promising for AAV enhancement overall. Also learn how CADs and AAV capsids may escape endosomes by disrupting bilayers, and we'll learn which AAVs and CADs destabilize membranes the most. So again, we're learning which might be most promising. And in my last aim, we'll learn where CADs change the endocytosis of AAV capsids, and learn which steps of endocytosis CADs seem to damage most. So overall, this will help us learn how CADs modify AAV gene delivery and how we can exploit those modifications. Wow, that was way better than I thought. Okay.